Hi, it's Kelly Shanahan, board member of Metaviver, here actually live and in person with Dr. Chuck Harrell, who received one of our grants back in 2014. Chuck will tell us in just a second. I've been vaccinated, Dr. Harrell's been vaccinated, so it's cool that we don't have our masks on. So I'm going to turn around and we'll say hi to Chuck. Hey, so tell us a little bit about when you got your grant from Metaviver and what you did with that money we gave you back in the day when we weren't giving as much money as we did now.
Very true. That money was fantastic, and I had just gotten here to BCU and had just started my own independent research laboratory. And that grant was to fund a liver metastasis project. So we used that money to develop some novel models that would allow us to study.
Hey, hang on one second. We got a helicopter going by here. Okay, the helicopter went away, so now we're back.
All right, so with that money, we were able to develop a novel set of patient-derived xenograft metastasis models. And so what those are patient samples that we get from the clinic that we can then put into a mouse and transplant serially so that we don't have to grow those cells on a plastic dish. So it kind of keeps the normal patient environment intact. And so what we did was we learned how to seed cells in the liver so that we could then track how well therapeutics would work in the liver in real time. And so specifically what we did was some genomic or genetic testing, and we identified differences in these cancer cells when they were growing in the liver as compared to growing in the mouse mammary gland.
So I think PDXs are seriously cool, and obviously we can't study everything in human beings, and so I think using these PDX models, which again are human cancer cells transplanted into a mouse model, are really awesome. And one of the things that Metaviver is hoping to do with our RFP, our request for proposals for the 2021 cycle, is to work on the development of newer and expanded models. So, and also a more diverse group of patients from whom these models are derived. So I'd like to ask you about, first of all, how many different patients were used roughly, and was there any racial diversity in the patients who were used in these models?
Absolutely. So initially we acquired a set of 14 patient-derived xenograft models. They were not very genetically diverse. I believe we had one African-American patient. Most were European ancestry and a couple Hispanic ancestry. Since then, we have acquired 6 additional African-American derived models that we're heavily investigating right now, and we're setting them up in similar experiments like we did with the Metaviver studies back in 2016 and 2017.
And what about the subtypes that were used? Because it seems like there are better models for certain subtypes versus others. So what were your liver met PDX models?
Initially, we were focused on triple-negative breast cancer primarily, though we also did establish one model of a HER2-enriched subtype and 3 models of luminal B or ER-positive breast cancer. We're now up to 23 models, and we have, I believe, 6 ER-positive, still that one HER2-positive, and the rest are triple-negative.
Okay, that's good that there's, because clearly, as we all know, the different subtypes make such a difference. So what kind of things have you guys found from using these models over the years? And what year did you get your grant? Do you remember?
It was funded for 2016 and 2017.
Okay, so you were our 2015 grant cycle.
The Abby Rappaport liver metastasis grant, yes. So one of the big things that we did was we identified genetic pathways that were differently activated in liver metastases. So that meant when the cells were growing in the liver, what was promoting their growth. And we found that the SARC pathway, which is downstream of the EGFR, was highly activated in metastases. At the same time that we were doing these genetic studies, we wanted to really figure out the best treatments, because we can do all the genetics we want, but if we can't apply those genetics to the patient, then we're not really doing what we're looking to do. So what we started to do was to take those PDXs, digest them into single cell suspensions, and put them in a dish and do high throughput drug screens. And so we started to do test 1,300 different FDA-approved drugs, and we identified a set of drugs that were highly effective across many of the triple-negative models. And interestingly, many of those effective drugs were also EGFR inhibitors. So we had data from our in vivo studies that said targeting EGFR may be important to stop liver metastasis growth. And then in our drug studies, it also said, yes, we can kill the cancer cells with these EGFR inhibitors. So we're very active in trying to target EGFR, but not as a mono agent, because EGFR inhibitors have been tried in the clinic and they have largely failed as single agents. And so we're trying to find a combination, a second drug that would work well at the same time, that would give an additive or a synergistic effect with the EGFR inhibitor.
Yeah, it does seem that combination therapy for a lot of our cancers is going to be the way to go. You got to close the door and the window in order to keep the cancer under control. So, you researchers in the lab come up with these great ideas and these great models, and you experiment on them and you find all this stuff, and sometimes drugs are found and the drugs are great for a while. And then inevitably it seems that resistance develops. So is there any work you're doing to try to combat resistance or figure out why resistance happens and what we can do about it?
About 3 years ago, I was working with a patient advocate who had triple-negative breast cancer to her liver and lungs. She initially responded very well to chemotherapeutics, but eventually the lung metastases came back and she no longer responded to chemo. She ultimately passed away from lung metastases. So seeing that, we really wanted to develop those models in the lab. And so we took and tested all of the triple-negative breast cancer PDXs that we had for for how carboplatin, a chemotherapeutic, would affect them. We found that some of the models, it didn't matter at all. They just kept growing. They kind of ate it and they kept growing through it. Whereas others would drastically respond, and we could actually totally cure some of the mice with carboplatin. But
remember, people, humans are not mice. Life would be a lot easier in some respects if we could scurry around and eat crumbs, and all these drugs that look promising in the lab would work on us. But we're not so simple. Go ahead, Chuck.
That's right. So we really wanted to develop some carboplatin resistance models. And so we did some suboptimal treatments of some of these PDXs that were initially very responsive to carboplatin. And we would do one dose of carboplatin, the tumor would shrink down, then we would let it grow back. And then we'd hit it with carboplatin again and it would shrink down, and eventually it stopped shrinking and it started growing right through it.
It's kind of like, no offense here, but for those of us that are married to, and or have, even better, have kids, and the whining is sort of, we tune it out. It seems like our cancers do the same thing. You expose them to the drugs enough and they begin to tune them out.
So what we're doing at this point in the lab is we're trying to develop many of these acquired carboplatin resistance models, and then ask what is different about the original tumor versus the carboplatin resistance tumor. And so this is involving a lot of genomics, a lot of RNA sequencing, and more recently we're doing a lot of single cell RNA sequencing. That means looking at the genome of every single cell within maybe 5,000 or 10,000 cells to see if there's different populations that emerge that promote the drug resistance.
So, I think a lot of us have heard of RNA now, thanks to COVID and the mRNA, messenger RNA vaccines. But this whole idea of single cell sequencing sounds super, super cool. It sounds super expensive too. So can you tell us a little bit about that technology and how it's made such a difference in research over the past, I don't know how long it's been used, but...
Yeah, probably over the past 5 years is when this technology has really emerged. We acquired a 10x Chromium single cell RNA sequencer about 2.5 years ago. It is about 10 times as expensive as normal bulk RNA sequencing. But the best analogy I can give you is if you were going to make a smoothie and you blended all of your fruits up, that would be your normal RNA sequencing that occurs, and you get one taste. When you do single cell RNA sequencing, you're eating each piece of fruit individually, and you can taste each bit of fruit and learn about each bit of fruit. And so what we can learn with this single cell technology is maybe when the carboplatin resistance tumor starts to grow, maybe a lot of grapes or a lot of blueberries are present within the smoothie, and we can really see that. So we're really learning about the different cell populations, and since we have that information, we can try to find drugs that will target a population as it emerges during drug resistance.
That really sounds like it's going to make a big difference, and your analogy is just spot on. I really could see that. You know, so much of the scientific stuff is challenging to understand, and I, for one, am a big fan of analogies, and that's an excellent one, and I may steal that sometime.
Very good.
I've
stole it from somebody too.
Okay, yeah. You know, why reinvent the wheel if we don't have to? I'm going to pause. So with drugs and drug development, it can be like reinventing the wheel, you know, coming up with something totally new, versus, you know, the whole reduce, reuse, recycle. So can you tell me a little bit about the drugs you're studying? Are they concepts that are not yet approved, or are you looking at already FDA approved drugs and seeing if they have a place in the armamentarium of the treatment of metastatic breast cancer and other cancers?
Sure. We're doing both. Primarily, we're trying to use drugs that are already clinically tested or FDA approved. So the drug library that we purchased is an FDA approved library. Because the goal there is, you know, if we can find something that works well in a dish, in a mouse, then we can immediately translate it to the patient. But with that said, we are working with chemists here at VCU that are making brand new novel compounds that we can then test and kind of do more exploratory type of studies.
Okay, great, thank you. I love the idea of using things that are already out there, because clearly if there's already safety data and there's FDA approval, it gets to us patients a lot quicker that way, and we certainly need everything we can get. Earlier you mentioned with one of your projects you were working with an advocate. That's one thing that I personally think is very important. I use the slogan, nothing about us without us. And it's one of the things that for the 2021 grant cycle and moving forward, that Metaviver is going to require that there be an advocate. And my preference, and certainly, you know, one of the things that I emphasize is don't like call us 3 days ahead and just ask for a letter of support. So can you talk a little bit about how you use advocates in your research?
Sure. We've had, I think, 4 or 5 different advocates that have worked with us with different levels. Some want to come into the lab, they want to see what we're doing. Others want to read some of the grants that we're working on. We've definitely used them to help us write lay abstracts. So when they see the scientific aspect of our grants, how can we translate that such that my mom or my grandma could understand it? So that's one of the reasons and the best ways that we've been able to work with them. But most importantly for me, I'm really trying to model the disease as best I can in the lab. You know, we're working with mice, but you know, when we learn about somebody's treatment approaches and the toxic effects that they had, and then what drugs they got switched to, we try to model that in the lab from day one. And so that's for both ER-positive disease and for triple-negative breast cancer.
Yeah, and I'm so glad to hear about you using advocates. One of the things that we've learned with COVID is there's too many meetings in real life. You can Zoom a lot of things, and that you don't necessarily have to be in the same geographic location in order to utilize advocates. And certainly if there's ever a way that Metaviver can help, if you're looking for someone with a particular subtype, you know, we've got a great database of people, and we would love to do that. There are some organizations that are starting to put together databases of advocates, because I think that by working together, we help each other, especially basic science bench researchers like you. That you're not in a clinic, you're not seeing patients, and I also think it's important because what I hear a lot is, how come we haven't cured cancer yet with all the money and time? And part of it is what you do is super hard, and you can't run a marathon before you've learned how to crawl. And in terms of cancer research, I think we're still in many respects at the crawling stage. We don't understand why metastases happen all the time. We don't understand how resistance to our drugs happen. And we got to figure that out before we go, throw a bunch of meds at it. I'm hoping we get an Operation Warp Speed for cancer research, because one of the things that we have learned in 2020 with the COVID pandemic is that if you throw a lot of money and you make people cooperate and share data, that we can accomplish a lot in a relatively short period of time. So I'm kind of hoping for a cancer Operation Warp Speed, and Metaviver, to put that in perspective, what we are able to give in grants is pennies, but it can be pennies that really start an idea and really get things moving.
Absolutely.
Yeah.
We would love that.
And just to remind you that you, just because you got one grant doesn't mean you can't get others. And we have been able to, thanks to the generosity of our supporters, been able to up our grants. I think you had an early career investigator grant, which at the time was $50,000, and we're up to $100,000 for that 2-year grant now, which I think means people can get a lot more done. And if supporters that are watching this continue to donate the price of a cup of coffee, etc., then we'll be able to increase those amounts or fund more research in the future. We were able to fund $4.4 million in 2020 in the middle of a pandemic, when other bigger, pinker organizations didn't fund any new research. So we're super proud of that, but we're most proud that we have incredible people like you who've done amazing work with the seed money that we were able to provide.
Thank you very much for your support.
Thank you. Hey, so I'm in Dr. Harrell's lab right now, which is really cool and way different from the labs when I was in college. And so hang on one second and we're going to get another update. So this is, you're doing drug sequencing right now. So tell us a little bit about what's going on here.
So what we're doing right now is we are testing 96 different drugs, and to see how effective they are at killing or slowing the growth of a single patient-derived xenograft. So on the left is the different drugs, the 96 drugs, and then on the right are where we have the cells in the wells, and so they're each being treated with a different drug.
Wow. And that's all automated now, and you used to have to do that by hand, right?
That led to
That's
many sore
crazy.
shoulders
Yeah.
for many people.
And so now it's picking up a different.
That's right, row 2 of
Uh-huh.
the drugs. So this is a different set of 12 drugs, and by using this robot, it makes things a little more efficient and more consistent.
And more precise, I would
there's
think,
less
too.
operator error.
Yeah. Wow, that's so cool.
Hi, I'm Jackie Gribble. I'm a PhD postdoc now. I'm no longer a PhD student.
But you're still doctor.
I am a doctor. I'm Dr. Gribble. So in Dr. Harrell's lab, I have 2 projects right now. The first one, I'm looking at drugs that could potentially work to benefit tamoxifen treatment for patients with ER-positive breast cancer. And then for patients with ER-negative breast cancer, I'm working on a racial disparities aspect, working with samples that are much more racially diverse than what has traditionally been used.
And I am so, I may be old and I may be white, but it is so good to hear that there is research being done that is more reflective of the population who actually has breast cancer.
Sure.
We know from research on, let's say, aspirin in cardiovascular disease that all that data that was extrapolated from men didn't really work in women.
Yep.
And so to to find that someone's doing rigorous science around whether there are any racial or ethnic differences that can be used to better treatment, because we've got to do something to address the 42% difference in the mortality rate. So thank you so much for the work you're doing.
For sure.
And then you can apply for a Metaviver grant too.
I certainly will.
All right.
Hi, my name is Timothy Smith. I'm a member of Dr. Harrell's lab, a postdoc, and my project is having to look at carboplatin-resistant cell lines and try and target pathways to overcome that, and drug synergisms, drug combinations that will have a synergistic effect.
Yeah,
to overcome that.
we need to overcome this resistance,
Yeah,
you know?
exactly. Yeah,
sometimes we need to be the resistance, and then sometimes we need to overcome the resistance, especially when it comes to treatments. So what is this cool looking colorful thing? Are those VCU colors?
Actually, almost, actually. I think it's more black and gold, but these are single cell RNA-seq data, so each one of these dots is a cell, and we're just looking at the gene expression of each of the cells to see if there's any differences between the chloroplatin resistant lines and the parental lines.
So what is the difference in the colors there? Does that signify anything?
Yeah, so these are actually just the different clusters. So the blue here will be the chloroplatin resistant cells, and the orange are the cells that aren't chloroplatin resistant. Okay. So, and then you can look at the differential gene expressions to see the genes that are either upregulated or downregulated.
Cool.
So,
Thank you.
yeah, no problem.